Hi, everyone. So it's 10:45. We will start the session now. Welcome, everybody. Just a few housekeeping messages for the speakers. This is a microphone. We will ask you to hold it. It's not for the room, it's for the people online and also for the translation. If you need translation, there are QR codes in each row. So you just pick one and scan the code. And my name is Lina Estupiñán Suarez. I'm a Colombian researcher working at Germany for the German Center of Integrative Biodiversity Research, and I'm part of GeoBond. I'm co-sharing the session with Andrew Rodriguez. Andrew works in GBIF. This session has two parts. Part one is today, part two is tomorrow, the same time, 10:45, in the same room here in Tolima, okay? So we will just want to give a short introduction of this session, Beyond 2030, building the data foundations for biodiversity action to support the Kunming-Montreal Global Biodiversity Framework. So to start, we know that the Kunming-Montreal Biodiversity Framework is already in place for almost 3 years. We have 23 targets for 2030, and we have 5 goals for 2050. Some of these targets are more related to biodiversity assessment, restoration, monitoring. Others are about sustainability and how we can have development with also have preserving nature. And the last ones are more focused in the biodiversity infrastructure and collaboration. In this session, we are also mostly focused on target 21, that is about the data mobilization and how we can ensure the base available data, information, and knowledge that are accessible to decision makers. This is the overarching umbrella of this session. But of course, we are going to look to other targets like 1, 2, 3, and 8 for restoration, assessing ecosystems. So we will have a mix of presentations. Some are more focused on the first target, and others are more like focused on the target 21. So we will have 6 presentations, and then we will have 7 presentations, I'm sorry. We will have 15 minutes for each presentation. We have 20 minutes presentation and 3 minutes for questions. Our room support, it's behind. She will show the presenters when 5 minutes are left, and then the last 2 minutes. So you just need to look behind. She's telling you how much time you have. And just the last information I want to provide is like for these 2 sessions, this part is focused more on this global perspective about all these tools that are available. And for tomorrow, we have a smaller or shorter talks that are more focused into national and regional experiences. With this, I want to introduce our first speaker. So can we, ¿puedes poner la otra presentación? This is for you. This is for the slides. You just find the root. So we start with David Patterson. He will talk about mapping what matters, foundational ecosystem data for the GBIF and beyond. So welcome.
No, it's okay. I think we're good, yeah. Right, thank you. My name's David. I'm from GEO, the Group on Earth Observations. Today I'm going to talk about the Global Ecosystem Atlas. I'm the project manager for this initiative. I'm going to talk about 3 things. I'm going to talk about what the Atlas is, and then I'm going to talk about technology and how we're doing it. Then I'm going to conclude about where we're going next. It should become quite clear how this fits into this session today as we go through the talk. So the Atlas, we are trying basically to put onto the map the world's ecosystems. We're trying to make sure that that data is open, accurate, and up to date. We are trying to put it into one standard, one typology, the IUCN GET, and we're working a lot with countries to get that data in. This is what, oh, goodness. So this is what it looks like as its beta version. You can go online, website's live. So the product we're making is it's a 100-meter raster, and it's at level 3, so it's 110 classes. So a land cover map would say 10 to 20 classes. 110 is obviously quite technically difficult, and I'll come on to how we're doing it in a little bit. We will be updating this. As you can see, there's obviously a lot of grays on the map. We will be filling that in, and the aim is to have this completed by Q4 2026. So obviously a lot of work to do. This work is being led out of GEO in Geneva. Our technical partners is James Cook University, led by Professor Nick Murray, his Global Ecology Lab. But we're also working with a wide range of partners to make this project happen. I won't linger on this too much, but obviously this data is a foundational product. We're not trying to replace anyone's work. We're just kind of trying to tidy up the filing cabinet and get all the data organized and available to use. So it's kind of like trying to build Microsoft Word and let people go off and write whatever they want to in those documents. So obviously the data can be used for countries, for CVD reporting, can be used in nature finance, companies themselves or FIs or investors, obviously can be used by science. Everything we do is fully open. So all the data we produce, all the code we produce, all the training data sets we produce, as I'll come on to, will be fully openly released. So just to go over that again, so Atlas is a collaboration with CBD and IUCN to get at the core of ecosystem extent for GBF GoA. It's a public good, and we are working to kind of get together the existing data and fill the gaps where data doesn't currently exist. So good stuff. We'll talk about the technical stuff now. So there's two ways data comes into the Atlas. On the top row, we have AI efforts. So about 30% of the map will be generated using AI approaches, which I'll come on to. And on the bottom row, about 70% of the map will be from existing maps, which we crosswalk into them. So if we consider this existing map, first of all, we're working hard to build a catalog of existing data sets. We're up to 355 existing ecosystem relevant data sets. There's an evaluation process where we look at each data set to see if it's fit for purpose. There's a selection process. And then, of course, it might not be in the right typology. And so they cross-reference that to the GET. It's a whole process involved in that. IUCN have released a framework for that. And then there's a geoprocessing process to put it into the right projection, etc. And then that's ready to go into the map. So to get this data, we're spending a lot of time talking to countries. We have staff working specifically on this. So on the map, you can see the red is existing data from the proof of concept, and the blue is data coming in shortly. We'll be doing quarterly updates, hopefully from middle of next year, to get some of this data out earlier. Data coming in from US, Australia, EU, and Botswana. Interestingly, some countries have approached us, like New Zealand and South Korea, and they're working to develop maps which fit with us. And we're also working with other projects in this space, like ESA Weed. We're developing, I think, 6 national maps this year with countries and 5 next year. And we'll integrate those maps when they come along as well. Oh, goodness. Sorry, guys, this is very delicate. Can you go backwards here, please? Okay, yeah, no, stop. And one more. Yeah, okay, so on generating new maps, this is done using a big training data set. We go to the next slide. Okay, So this is the training data set we're building up. We have a team in Australia, eight annotators working on this. This is 90,000 points, which are covering as many ecosystem functional groups as possible. There's 110 ecosystem classes we're working to. And this is where we're currently at. So obviously, it's a lot of work. We will be releasing this training data set, hopefully at Q4 2026. So then once we've got those two data products together, we've got the new maps, we've got existing maps, we push them together and then they come out. There's a whole complex staging process, etc., which we don't need to get into today. The other thing we do is we do national accelerators. So the two ways we get data in primarily is that, existing maps and new generated maps, but we also work with countries who don't have existing maps to help them kind of get kickstarted on the process. So we just finished capacity building in the Maldives, where two of our field staff went out with a team of 6 local staff and collected data from the Northern Atolls, which is really great ground verification data. Okay. So currently we are on track to deliver the whole map by Q4 2026. We have the core technical pipeline behind all of this. You saw the annotation, the training data set is well underway, 14,000 points done. We have 355 ecosystem data sets cataloged, and we're working now on building out the new website to house all of this. And yeah, as I said, everything's going to be made open. So where next? So this project is funded for its first phase. Phase 2, which is 2027 onwards, we're looking at things like changes in ecosystem extent, pixel change detection. We're looking at moving to higher granularities in the IUCN GET, so we've been moving down to level 4. So we're currently working with 110 classes, which consider function, but they don't consider composition. So when you move down the typology level 4, you end up with a lot more classes. And then there's the problem which everyone, in my world, everyone's talking about, which is ecosystem competition. How do we get at that? We're keen to kind of support and help in any way we can to help move forward in that space, but obviously incredibly difficult and challenging. How you can get involved, please come speak to me if the project sounds of interest. We're very interested in people who are existing ecosystem data sets, interested in anyone with training data sets, also very interested in the marine realm, if anyone's got data or thoughts about how we might get better mapping ecosystems in the marine. But I think that's everything. Thanks so much.
Yeah. Thank you, David. We have time for questions. So is there questions in the room or in the chat?
Hi, thank you for your presentation. It's really important work. I was wondering about the training data sets. I'm not quite familiar with the ecosystem data standard. So the training data set, you had a lot of points. I was interested in Canada and Quebec points. So I was wondering what type of points are they? Are they GBF points or?
One second.
Yeah, they're just random sample locations where our team goes and works out what ecosystem's there. And then that data set gets applied to foundational models, AIs and Google's currently. And then that generates what the foundation models thinks there. So it's basically training data for AI. Oh, yeah. So there's a whole structured process. So yeah, it's a very involved thing, but yes, I'll talk about it offline.
This question is about the URL, and it looks like it's getting an error. I don't know. I think you can check online.
Just
yeah,
Google it, find it, yeah.
Yeah.
Be fine.
All right, so thank you, David. So we continue with an online presentation. It has been already recorded by Matthew Vickers from CSIRO in Australia. The time difference is large, so he's not online for questions, but we can present the video now. His presentation is on applying continuous community-level biodiversity indicators for CBD reporting. So here we have it. All right. I think we just need to wait until it can be played for people online. Okay, we just need some volume. Okay, yeah, this like technology is very helpful, but also very challenging, especially when you have these hybrid events. Yeah, we just wait a couple of minutes. Okay, I think maybe I will just, yeah, okay, we can hear something. Can you be louder? I think that's good. Okay.
We hope the government use the results, regulation, indicators of the government, track on it. When it comes to measuring biodiversity, indicators must be spatially explicit, but indicators that are spatially parallel, limited comparisons. We need indicators of scale, capable of aggregation, flexible classification. Data must be forced, the limit. And model built on this, and this spatially explicit patterns or responses. Indicators and expert judgment, specific cases is reproduced. Typical direct indicator. Indicators, units. We need to be measuring by directly changes in land cover species. No. Indicated meaningful directly. And this is where the bill using notation, we have only the indicators of global biodiversity. It is pure. Really very long geographic modeling, large scale biodiversity. It's just as important to know that in real life, the building nature could fail you, in Australia. Therefore, we're building indicators, each addressing a different dimension of biodiversity. BHI, the Biodiversity Habitat Index, tells us the amount of biodiversity likely to persist in the future given how we evolve the ecosystem. Very, the Biodiversity Ecosystem Resilience Index tells us how resilient is climate change. We have two compartments: protected area representativeness and effectiveness. Protected area representativeness tells us the degree to which protected areas capture the full range of biodiversity and ecosystems under assessment. Protected area connectedness, on the other hand, indicates the degree to which species are affected by the together these baseline process, whether that support by change. The building indicator. There's true specifically, there's one goal. BHI, biodiversity is enhanced or restored by ecosystems and sustainable species abundance. By using indicators, scenario testing, spatial planning design, BHI and Berry both address type 1. BHI and Berry also address 2, tracking progress in and providing a mechanism to test whether methods are likely to enhance or hinder climate resilience. The park indicators are designed to analyze protected areas for target 3. Park R and Park C can be used to test whether protected areas contribute to the 30 by 30 target in a meaningful way that helps us reach our ultimate goals. Not just conserving blindly 30% of the land, but conserving the right 30%, the 30% that gives us the best chance of survival. And through its integration of habitat quality and climate change, Berry addresses target 8, estimating whether species and systems will persist under climate change. BHI is the indicator that underpins the others in Bilby. BHI uses generalized dissimilarity modeling to create and assess biologically meaningful assemblage-level biodiversity patterns. We can compare the community at the blue site here with the communities at each of the red sites to gain a measure of biological dissimilarity, which is the complement of similarity. We relate that biological dissimilarity to changing landscape parameters such as climate, soil chemistry, terrain, land cover, and distance. The key is that GDM fits nonlinear functions, which means that the GDM can simultaneously account for 2 nonlinearities. The first is a nonlinearity in how community composition changes with environmental distance, or similarity in species assemblage composition. Similarity in species assemblage composition tends to decay with geographic separation due to limits in biological processes like dispersal capacity or other challenges arising from biogeography. The other nonlinearity is a relationship between compositional dissimilarity and environmental gradients. It's nonlinear because, for instance, some species respond gradually to changes in temperature or soil chemistry, while other species shift very sharply at given thresholds. Here we see 2 sites that have fairly similar habitat attributes and response as our blue circle, while the third is quite different. GDM models the environmental gradient with an I-spline smoother and retains a separate nonlinear geographic distance term. Taken all together, the result is a biological distance surface. It's essentially a multi-band raster map that predicts expected turnover in species assemblages between any two locations on the map. And this is what lets us move beyond species counts to a much more powerful estimate of assemblage-level biodiversity patterns. To assess habitat condition, we then combine these biodiversity surfaces with spatial data on land use and land cover quality. Globally, this comes from harmonized land use data sets and satellite products, though specific use cases can introduce their own local data to address particular needs. Together, GDM and habitat condition mapping give us the machinery to estimate how much of the original biodiversity is expected to persist and how resilience is changing under land use impacts and climate change. BHI is the indicator that underpins the rest of Bilby. With its generalized dissimilarity models, it maps compositional turnover of species assemblages. Turnover combined with the global land use data gives us BHI, and this is mapped for each grid cell as an estimate of the proportion of species expected to persist indefinitely. BHI results are scaled from 0 to 1, and BHI is spatially explicit and reportable at any scale, from individual properties to states, regions, national levels, or even global. A BHI of 0.7 means that on average about 70% of the originally occurring species are expected to persist into the future. Comparison of BHI amongst varying scenarios can provide decision makers with the information required for better spatial planning, addressing target 1. Tracking across BHI across time can be used to assess performance in restoration, addressing target 2. BERI, the Biodiversity Ecosystem Resilience Index, builds on the concepts of BHI by adding climate change. We calculate climate analog trajectories, indicating where a cell's future climate occurs spatially. Here you can see the orange cell moves in space as climate change progresses. If there's continuity in the connectivity between the original cell location and where it's moved to under climate change, the cell will score a high BERI score, indicating species can respond to climate change by tracking the trajectory. If the focal cell loses connectivity along the way, or it ceases to exist, it receives a low BERI score, indicating a low resilience to climate change. Governments can use BERI to report on target 1, 2, and 8, and more locally, an organisation could use BERI to prioritise restoration that reconnects climate refugia, boosting species climate resilience across a region. For park representativeness, we overlay protected areas from the World Database on Protected Areas with our GDM-derived biodiversity units. For each unit, we calculate the proportion of biologically similar habitat under protection, and then we summarise how evenly that protection is distributed across biodiversity units. High Park R means that all the ecosystems available are well represented in protected areas. Low Park R means there are gaps in the protectedness. For Park C, we treat protected area grid cells as nodes in a network. We calculate dispersal probabilities between nodes. We use geographic distance and resistance surfaces, and then apply metapopulation connectivity concepts to calculate connectedness. High Park C means a well-connected system of protected areas. Low Park C means our protected areas are isolated fragments. The park indicators are to be used to assess the amount of protected area in a way that's more meaningful than hectares alone. We can use park to test whether the area under protection is representative and connected. Protecting a randomly or poorly chosen 30% of the land will not yield the results we need. We need to be deliberate and effective in our choices of which land to protect to meet the 30 by 30 target. Park indicators are designed to help ensure protected areas occur where they're most needed. So we can use Park C to communicate the effectiveness of connectivity to governments, and they can use the information to actually strengthen the protected area networks rather than just adding isolated fragments. The Bilby indicators can be accessed in our portal. There you can browse globally or by country for the indicator of your choice and download the data. This link and the QR code takes you to our Bilby indicators dashboard, which the next slide will take us for a quick tour.
Here is our Shiny app dashboard for the Bilby indicators. At the top, we can select a global overview or individual country detail. We select an indicator from the list, and we can see that from 2000 through to 2020, there's been a global decline in BHI with what almost looks like the beginning of a flattening of the curve here. We can select the final year for analysis over here. Further down the page, we have a bar chart with one country per column indicating the net change in the indicator BHI from 2000 to 2020. Most of the countries here are shown in red, indicating that the BHI value has decreased over the time, though some are in blue, indicating a gain in BHI since 2000. At the bottom of the page, we see a global overview with BHI aggregated at the country level. We can change to the country detail and again select an indicator from the drop-down list. Here we have park representativeness for Australia. Park representativeness is shown, or these indicators at the country level are shown, broken down by WWF biomes, with a whole of country summary also overlaid. You can see that since 1970, Australia has gradually gained in park representativeness. We can select any country from the list and to see that in the case of South Korea, also there has been an increase in park representativeness. Here we see the net change in the index since 1970, broken down by country. Again, one column per country, and in this case, we see that every country in the world has gained in park representativeness since 1970, with our focal country shown in orange. At the bottom, we have a spatial representation of the indicator. You can slide the transparency slider to overlay the indicator on the map, and the map is available in various languages and formats as you might require. Here on the left, we can download the chart data, and at the very bottom is the Contact Us button. Please get in touch about our Bilby indicators.
4 Bilby indicators were accepted as component indicators under the Kunming-Montreal Global Biodiversity Framework. The indicators are spatially explicit, reproducible, and globally consistent. Individually, each indicator measures a different facet and together provide a coherent interconnected system. BHI tells us how much biodiversity we have left. Berry tells us whether the habitat and biodiversity can persist under climate change. And the park indicators tell us if our protection system are representative and functional. Thank you for listening to my talk today. I hope you find our indicators useful, accessible, and easy to interpret.
And actually also, I'm curious about how many people here are working with the governments and doing the reports. Okay, that's good. We have some people. Okay, so that will be very good for the discussion. We move to the next presentation. This is by Sidra Ikram, and I think it's a joint presentation, if I'm right, yeah. They come from Kew Botanical Garden in the UK, and they will talk about bridging biodiversity knowledge gaps, unlocking botanical and mycological collections at Royal Botanical Gardens, Kew. So welcome. Yeah, that's fine. Okay.
Good morning, everyone. My name is Sidra Ikram. We've come from Kew Gardens today. We're talking about our mycological and botanical collection. Next slide, please. Yeah. Louder? Okay, sure. So we know that some of the biggest threats to biodiversity includes climate change, food insecurity, and biodiversity loss. And the target 21 of the KMGBF emphasizes using the best available data that we have in order to support the effective management of this biodiversity. Next slide, please. At Kew, we have 12 major scientific collections, including seed collection, living, spirit, and economic botany. And although we do have one of the largest and most diverse mycological and botanical collections, we've not been very good at making this data accessible for the world. Currently, only people who come to Kew can look at the specimens, or if you can request a sample, but it's not very accessible and it's not very environmentally friendly if people are having to fly from all over the world. Next slide, please. So in 2022, we started our digitization program. It's a 4-year program fully funded by the UK government, and it has 3 major components. The first is the digitization, which is imaging and transcribing. So we're only doing our herbarium and fungarium specimens for this project. It's about 8.5 million specimens. The second part was to basically create a central database, because all of our different researchers and science collections teams had their own separate Excel sheets and databases. So we've made what we're calling the integrated collections management system. So it's a central database for all of our data to come together. And the third and probably the most important part is to get the information out there. So we've developed a data portal. It's our online platform where we're hoping to get free and open access for not only researchers and scientists, but the whole general public. Next slide. So it's a little bit of a clip of how we do our imaging work at the stations. We have these sort of light boxes set up, and we use barcodes and scanners. So as of the 10th of October, we have imaged 7.9 million specimens. We've transcribed 5.5 million of them, and we are on track to finish the project by the end of March 2026. Next slide, please. Talking about our ICMS project, we actually completed that earlier this year in March. So we've used a company called Earthcape, who are based from Finland. And with them, they've helped us sort of customize their existing software for us. It's a little bit bespoke. And we've been able to get all 12 of our major collections on there together. Next slide, please. And our data portal, we actually launched it earlier this year in April. We are still going to call it a beta version because we're still improving it. And to date, we have 6.71 million specimen records online. There is the link, the address there. Please do, if you have a chance, have a look and give us some feedback. We're still trying to improve it. And I think our next target is to make sure that all the images online are actually high resolution. At the moment, some of them aren't. I'm going to call Udi to continue the presentation.
Thank you, Sidra. So I'm Udayanganiliyu. I will highlight the key data challenges we face, our solutions, and how they help us to bridge biodiversity knowledge gaps within our collections. So integrating data from 12 separate collections into one system required standardizing inconsistent data practices. So we held numerous workshops across the Institute to establish shared vocabularies. Sorry, the next slide, please. Shared vocabularies, the data workflows, and standards. So we use widely accepted international data standards such as Darwin Core, Chadwick, gene banks, museums, and botanic gardens. So we documented all the process in detail. Now we have a modern system with consistent data. It works well with other systems and support data and also research across the collections. Next slide, please. So standardizing taxonomic names was one of the major tasks. We had a mixture of unpublished names, names with non-Latin content, names without scientific authorships, and invalid names, very many inconsistencies. There were variable nomenclature and contradictory taxonomic concepts across the collections, making data fragmented. So we preserve the original name without losing the context while mapping them to an authoritative framework. Next slide, please. So we use the World Checklist of Vascular Plants and Index Fungorum as our taxonomic name backbone. This solution enables us integrate searches, reliable reporting, and future-proofing our data. Next slide, please. So currently in ICMS, we have over 6.7 million object records. Currently, herbarium and fungarium records by far the largest, reflecting the scale and history. Then living collection and live seed in the Millennium Seed Bank and in vitro collections are comparatively small, but crucial for ex situ conservation. So altogether, this record covers diverse data types, collection strategies, all unified in one system. Next slide, please. So for the first time, we were able to visualize geographic footprints of all our collections on single map. Previously, spatial data was scattered, preventing a unified geographic view. Next slide, please. So our collection also covers a wide range of in situ data from wild habitat. So some of the I listed here, like the dates, the taxon occurrence, their biological characteristics, the provenance, habitat, population, ethnobotany, vulnerability of taxon, and many more. And unlocking these data help us to understand the population and ecosystem changes over time to support the conservation. Also, in addition to this, these collections have a vast potential to unlock trade data for fungi and plants. Also, during the curatorial and horticultural activities, these collections generate a vast amount of secondary data, which are useful for conservation. Next slide, please. So I would like to share a brief case study from the Millennium Seed Bank to showcase how combining collection data with biodiversity or ecosystem parameters, actually we can support addressing real-world conservation challenges. So Millennium Seed Bank is a long-term seed conservation facility for wild plants. Through a global network of partners, seeds are collected and conserved through the world's largest ex situ seed conservation program managed by Royal Botanic Gardens Kew. Next slide, please. So this map highlights the geographic footprints of seeds conserved at the Millennium Seed Bank, including those originating from biodiversity hotspots. So data serve as a powerful tool to identify geographic gaps and help us to prioritize our conservation efforts where they are most needed. Thank you. Next slide. So here we overlaid the geographic footprints of useful plants conserved at the MSB with global biome data. So this is an example by integrating taxonomic, geographic, and ecosystem variable, we can better understand the ecosystem or ecological diversity represented in our systems, our germplasm, and where the critical gaps remain. So finally, this highlights the bioclimatic footprints of threatened plants conserved at the MSB, and this highlight by integrating the biodiversity data with collection data as well as ecosystem data, we can better understand the conservation challenges worldwide. So this case study, I know it is brief, but it showcased the transformative power of data in conservation and how we can help the future conservation activities. Thank you.
Yeah, thank you for the sharing these new data sets that are available, and it was a very good exercise how to build up on the standards that are already in place. Are there questions? I think we have time for, we don't have time, but maybe you can, I think we just take one. So maybe you can approach them after the session. Okay, but in the okay. All right.
Don't worry, it's very easy. I see you use the index fungorium for fungi, but I want to know what do you think about Mycobank? I don't know what of both is more actually in the taxonomy.
Just a second.
Thank you.
So the fungal taxonomy, the index fungorum, also called species fungorum. Sorry. So usually Q has a contribution to curated. Myself, I'm more a plant side of, so I'm not in a place to guide you in taxonomic concepts. But we are happy to, follow up after the meeting if you wish to. Yeah, we can provide you more details. Yeah, thank you.
Okay, that's great. We move to the next presentation. That's a video by Sarah Hubner. She's from Smithsonian Institute, from the National Zoo and Conservation Biology Institute in the States. Sarah, I believe, is online. She couldn't come, but she will be available for questions. So we can start her presentation. Yes, perfect.
As you all know, robust data is critical for conservation, and ecologists have deployed sensors at an unprecedented scale to monitor wildlife. Among these, camera traps have become one of the most powerful and widely used tools, capturing detailed images of animals in nearly every ecosystem on Earth. Each year, they produce vast numbers of biological images and videos, not to mention rapidly increasing amounts of data from drones, satellites, and other remote sensors. Yet, despite this abundance, the quality and accessibility of these data sets vary widely. In particular, certain kinds of labeled images, such as those that capture behavior, remain surprisingly scarce and difficult to use for ecological research. Yet, as our shared conservation targets become more ambitious, timely and accessible data are crucial to measure progress and guide action. These global frameworks outline how we collectively monitor and protect biodiversity, and camera trap data directly inform many of them. The Global Biodiversity Framework tracks progress toward international conservation targets, and camera trap data provide on-the-ground evidence of species trends. The essential biodiversity variables define core metrics for biodiversity change, such as species abundance and distribution, things that camera traps measure especially well. The Sustainable Development Goals, particularly life on land, rely on wildlife data to evaluate conservation outcomes. And the CITES framework focuses on endangered species and trade. Camera traps help reveal how those species are faring in the wild. These data also feed directly into major biodiversity indices, the Wildlife Picture Index, which uses camera trap data outright, the Living Planet Index, which integrates population data from around the world, various IUCN Red List indices, and the Human Footprint Index. Together, these connections show how standardized, high-quality camera trap data strengthen our ability to track progress across scales, from individual species to global biodiversity goals. Of course, meeting our shared targets is impossible without sustained collaboration. To generate indices and inform international frameworks, we need compatible data, and that means coordinated global monitoring. Distributed camera trapping networks like Snapshot USA and Snapshot Safari already collect data using standardized protocols, allowing for meaningful cross-site comparisons of wildlife and conservation outcomes. This model has recently expanded beyond the US and Africa, with partners in Europe, South America, and Asia adopting the same collaborative approach. Now, our collective efforts are being brought together under the banner of Snapshot Global, a unified framework that harmonizes data collection methods, timelines, and analytical standards. By integrating these complementary data sets, we can move from regional studies to truly global scale analyses, revealing biodiversity patterns and long-term ecosystem trends with unprecedented scope and consistency. The next challenge and opportunity is ensuring that these data become lasting scientific assets. That's exactly what Smithsonian Wild was created to do, transforming distributed monitoring data into a curated, born digital museum collection, the first of its kind at the Smithsonian. We're creating a lasting, accessible archive that transforms individual projects into a permanent and cohesive scientific resource, leveraging millions of wildlife observations to gain deeper insights on species behavior, habitat use, and biodiversity through time. It's also a cultural record, documenting not just the natural world, but how our relationship with it evolves. This collection can help us see both ecological and societal change. Standardization and expert oversight ensure that the data in the collection remain consistent, credible, and interoperable across borders and decades. By preserving these data sets under Smithsonian standards, we safeguard them for the long term, ensuring today's records remain usable for future science and conservation. And finally, openness builds trust. Public access and transparency extend the value of this work beyond research, engaging broader audiences in biodiversity and conservation. Preserving scientific value isn't just about storage, it's also about structure. How each observation is organized and maintained determines how useful it will be in the long run. In Smithsonian Wild, every field observation is treated as a digital specimen, not just a single photo, but a complete record that includes image sequences, labels, and detailed metadata about the wildlife, the habitat, and the equipment that captured it. Thinking about data as specimens ensures each record can be traced, verified, reused, and updated as technologies advance and taxonomies evolve. To protect that integrity over time, we focus on a few essentials. Ongoing validation to keep image labels and metadata accurate, curation protocols that standardize data and metadata so records collected across continents remain compatible, version control to document how records evolve, and secure redundant storage and regular audits to prevent data loss or degradation. Preservation gives us stability, but the real value comes from what that stability enables. Because the images and metadata in Smithsonian Wild are standardized and traceable, they can be reprocessed as new machine learning models emerge, allowing us to extract additional information that wasn't accessible before. For example, we're applying vision transformers to improve wildlife and behavior identification directly from images. Models like CLIP can link text descriptions with visual data, opening up opportunities for behavior recognition through new approaches such as WildClip. Beyond individual detections, spatiotemporal models allow us to combine time series and spatial data, helping predict species distributions, seasonal behaviors, and responses to change. And we're moving into multimodal modeling, connecting camera trap images with acoustic data to recognize species not only by sight, but also by sound. On the infrastructure side, pipeline automation ensures the data remain ready for these applications, assigning quality scores for machine learning training purposes, converting metadata to the CamTrap DP format, and automatically checking for taxonomic updates. But even the best data are only as powerful as our ability to share them. That's why interoperability and open access are core to the Smithsonian Wild Collection. These principles guide how we connect with the global data community, ensuring Smithsonian Wild contributes to and benefits from a larger ecosystem of biodiversity information. By converting our data to the CamTrap DP format, we ensure that metadata are structured consistently with international standards so they can be integrated and compared across networks. From there, we're sharing data through GBIF to provide global access to our observations and connect this collection to other monitoring networks. At the same time, openness doesn't mean all or nothing. We're establishing tiered access, making some data sets fully public while maintaining controlled access for sensitive information or specialized uses like AI training. That balance between transparency and responsibility builds trust, not only among scientists and developers, but also with the public who benefits from this work. In the end, openness is only meaningful when it leads to real-world impact. The data we're building aren't just for analyses. They form a foundation for better conservation decisions from global frameworks down to field sites and local institutions. The cyber infrastructure behind Smithsonian Wild is designed to connect not just systems, but communities. Right now, the collection consists of camera trap data collected by Smithsonian scientists across our ecology and conservation programs, but it was built with the capacity and intent to grow. We're eager to bring in data from other institutions and research networks that share our commitment to open, standardized biodiversity data. We're building this network with partners across sectors, conservation technology innovators, data science organizations, and research institutions. Together, these collaborations help us scale from project-level data management to a global cyber infrastructure for biodiversity discovery. On the technical side, we're developing platform linkages that allow for the fast and reliable transfer of large-scale image data sets, so that the millions of images coming in from field sites can move seamlessly into the collection and be published to GBIF for maximum discoverability. But our vision extends beyond camera trap images. We plan to eventually host collections of biological videos, acoustic recordings, and genomic data. Smithsonian Wild is also designed to connect with other biodiversity monitoring systems like MoveBank and emerging networks for acoustic and environmental sensors. Linking these data sets gives us a more complete picture of wildlife behavior, habitat use, and ecological change over time. We're also aligning with national initiatives such as NAIR in the US, which is expanding computational access for scientific research. Integrations with platforms like these keep Smithsonian Wild future-facing, ready to leverage shared cloud, compute, and data resources as they grow. At its core, this is about creating a model other institutions can replicate, born digital biorepositories that are interoperable from the start, designed for collaboration, and capable of accelerating biodiversity science worldwide. The more our infrastructure connects across platforms and partners, the more powerful it becomes as a tool for conservation. Because truly, data infrastructure isn't just about managing information. It's about empowering the people and policies that sustain biodiversity. We're already beginning to see what that looks like in practice. At the National Zoo and Conservation Biology Institute, field data from Smithsonian Wild are informing animal care, breeding, and reintroduction programs, and our work in the zoo is guiding priorities for what and how we monitor in the wild. As the collection grows, it will help to refine our field methods, strengthen our species maps and habitat models, and improve how we assess conservation outcomes at both local and global scales. Ultimately, these connections create a feedback loop, where each data set contributes not only to science, but to our shared goal of sustaining life on Earth. Thank you for the opportunity to present virtually. Although we're disappointed not to be with you in person, my Smithsonian colleagues and I remain deeply committed to working with all of you toward our collective conservation goals. My contact information is here, and Roland Kays is attending in person if you'd like to connect about new international snapshot grids. I'll also be standing by online to answer any questions you have. Thank you again for joining me today, and I look forward to connecting with you as we continue building the future of living data together.
Very nice presentation on wildlife and also observations and the importance of harmonized data sets. If Sarah is online, can you let us know in the chat? Can you say something, Sarah, if you are online? In case she's online, okay. So we can take questions in the chat or here if we have one. Or are there questions in the room? And in the chat? No? Okay. So yeah, we can move forward to the next speaker. We have Adelola Teminepo. Is she in the room? Oh, is he? We have both. Okay. Okay, so she sent a video. So let's play the video. So Adelola and her colleague. Yes.
A conservation biologist by profession. So I'm going to be presenting on gaps on biodiversity indicators for ecosystem restoration monitoring. As we all know, ecosystem restoration monitoring has become a global priority, which is to prevent, halt, and reverse the loss of nature. Also, there has been the UN Decade on Ecosystem Restoration to scale up large-scale restoration efforts between 2021 and 2030. To accelerate this action, there has been several commitments from diverse initiatives. For example, we have the Bond Challenge, we have the AFR 100, and this ambiguous commitment are to scale up the restoration of globally degraded ecosystem. And in alignment, we also have the target 2 of the Kunming-Montreal Global Biodiversity Framework, which is to restore 30% of all degraded ecosystems. And everything seems to complement each other, including several SDGs, and all to achieve the goal of human living in harmony with nature. And to actually capture the progress of restoration, I mean, there are commitments, there are restoration being done, but we also need KPI that reports on restoration status and their impact, especially towards the decade and also for target 2. So the FAO is working as the custodian of the target 2 to develop the framework for ecosystem restoration monitoring that will report on global restoration progress for the UN Decade and target 2 of the KMGBF. So this has been developed. So within the framework for ecosystem restoration monitoring, we have 4 components. The first is the registry, where users get to document their restoration data and good practices. We have the geospatial tool for visualizing restoration data. We have a search engine that shares good practices and initiatives that have been published. And we have a dashboard, which is currently under development, that will show global restoration progress from diverse sources. So in reporting on the indicators for target 2, as we all know, we have the headline indicator, which FAO is the custodian to report on area under restoration, but we also have other components indicator within target 2. For example, to understand the proportion of land that is degraded over total land area, the Global Ecosystem Restoration Index, proportion of key biodiversity areas in favorable conditions. This means that restoration needs to benefit biodiversity, benefit community, benefit climate, but we are focusing on the biodiversity aspect. So beyond collecting data on area under restoration, we also need to understand the KPIs that really matters to report on progress on restoration sites. So for example, target 2 qualifiers, restore 30% of degraded land with this qualifier to enhance biodiversity, enhance ecosystem function and services, enhance integrity, enhance connectivity. But how can we really measure progress towards these qualifiers and towards these complementary indicators? So we really need to understand the indicators that will show us progress to some of this area of impact. So within FIRM, we leverage the Aurora indicator to capture indicator relevant for biodiversity. It also has other indicators. However, we have our challenges. Before using the Aurora indicator, we actually leverage on resources developed by FAO and other restoration frameworks to understand the indicators that matters towards restoration. A majority of the indicators were focused on SDGs. So we actually wanted more indicators beyond SDGs and indicators that shows local metrics. So that's why we use the Aurora indicator. The strength is that it really captured local metrics. However, it's not really sufficient for all ecosystem types. So we integrated this into FIRM for selection of indicator at project level. And what we realized is that 80% of countries actually lack the capacity to collect data and report for the KMGBF target 2. So currently, we have the Aurora integrated into FIRM, and the way Aurora indicator is developed is that it has teams, it has metrics, and it has units. So what we realize, for example, I can select biodiversity, and once I select biodiversity, it shows the deep purple color, and it shows me the metrics under it, for example, community composition, to document data based on species richness, species abundance, and the diversity as well. But what we realize is that 90% of the projects we have in FIRM actually select teams, but do not select the metrics or document any metrics for it. They actually left it. So it's very difficult to report progress just using the teams biodiversity, the deep color. Within the interface, it makes it beautiful, but there's no quantitative data to really capture the progress, the KPI for biodiversity. So this has been a challenge. So what we realize is that why Aurora indicator capture local data, local metrics data, it is too generic and it is not tailored to ecosystem specific needs. Also, we realized that the restoration sites, based on the data that are documented into the FIRM, different restoration have their own objectives. So it is very difficult. At the end, we have majority of users using the custom indicator to document their own restoration indicators. So it becomes very difficult because how do we report on the progress, the impact for biodiversity, the impact for community? How do we report this progress? So it becomes too generic and not applicable. And this is some of the work, the working group we have in the task forces on monitoring for FAO are trying to address as well. So for now, we also realize that indicators remain largely global because we also try to see other EBVs that are available on the global scale, but they don't really have the local, they don't have it tailored to local metrics or ecosystem types for now. So it's very difficult. So what we try to do is to run a systematic review of existing EBVs that might exist. And we use some of this keyword search, which give us a 1004 paper to really see what might be useful for us. So the papers are cut across top categories of field, from environmental to ecological, forestry, and other fields. across these SDGs and within this year in which they are published. So from the search as well, it cut across different ecosystem types, different continents as well. So we try to show the numbers of publication we found across these scales. So from some of the top indicators that we found, especially trying to look at the linkages with some international recognized publications and to see which one is mentioned across all of these publications. So we noticed like species richness and species abundance, it's actually top of it. However, it also cross-cuts different scales. For example, across the avians, the birds, the amphibians, the reptiles. So we really need a standardized indicators that can be mapped from the local scale to the global scales that are relevant to report on some of these qualifiers of target 2 and the component indicators as well. So currently, we are also trying to run more analysis to understand the classification of biodiversity indicators, assessment of the relevance of each indicators, and FAO is working across all of these thematic areas. So based on the insight from literature that we found, there is a strong academic support for EBVs, but limited integration into restoration monitoring practices. They are not tailored yet into local metrics or by ecosystem types. Also, most literature recognizes that current biodiversity indicators do not adequately capture ecosystem functions, integrity, or connectivity. So, and we really need all of these to report for target 2 qualifiers and also the complementary indicators. So we have all of these indicators. If we have component indicators, we have the headline indicators, we have the qualifiers. We also need KPIs that are relevant to monitor these indicators, and it should be relevant to different ecosystem types and also useful at the ground level, especially at the local level. So we also found some papers that also talks about biodiversity indicators should go beyond reporting on species abundance and population. So we also want to understand how all of these are relevant. What should we really put into consideration? And also linking the socio-ecological aspect as well to make it relevant for human and nature. So we are still trying to figure all of this out. This is what we found from literature. And from literature as well, we realized that major indicators are focused on terrestrial ecosystem. So only few sources propose monitoring indicators for marine and marine ecosystems. So this is a huge gap as well. And we also need indicators for freshwater ecosystems. So these are some of the gaps we realized from literatures, and more work still needs to be done and also to make it practical. So in general, without wasting much of our time, so the gaps that we found out is that we found global bias, weak local relevance, like there are indicators at the global scale, but it's not locally relevant. And also the data availability and fragmentation. Also, we need the indicators to be narrow set. I mean, it's amazing to know like there are indicators for soil, water, reptiles, mammalian, avians, and different indicators across even structure. But at the end, we just need tailored indicators that are actually relevant. So this is a huge gap for us in terms of the data that are submitted into FIRM and the literature we review. So we still need a methodology to be able to report on this. So like also lack of methodological guidance and quantitative data on the representation of socioeconomic dimension. This should also be linked to ecological data as well. Weak integration of remote sensing and field data. So I think what some of the people who leverage, who uses FREM also want is having spatial data that can actually support local monitoring. So if they don't have the capacity, there should be spatial tools that can support the monitoring of this indicator, especially for biodiversity. So limited uptake of the EBVs and not fitting into local monitoring. I think I already mentioned this. And some of the recommendations we have is to broaden biodiversity indicators and strengthen socioeconomic integration, promote standardization and interoperability, integrate multiple data sources, adopt and mainstream EBVs, develop methodological guidance, which is very important. If there's this framework out there, there should be a guidance on how to use it and how it can be tailored to local context. Also to foster cross-scale monitoring system and invest in long-term monitoring. So I think, yeah, this is just in summary, our message is like we need to report for target 2 headline indicator, but they are complementary indicator, they are qualifiers, and there's not been good methodology out there that can be tailored to capture the KPI that are necessary to report on progress for these indicators. And we also need global EBVs to be tailored to local metrics and spatial tools that can support countries to build their capacity in terms of reporting on biodiversity indicators. So thanks, colleagues. Thank you so much. Thank you.
Using the indicators that we are proposing, how we can go from this global scale to really a national scale for reporting and the gaps and the KPIs that need to be reported. And I think that's actually really broadening a perspective of how we can really apply what we are doing. So with that, we move to the next talk. We have Florencia Gatarola. She's coming from Uruguay, but is based in Czech Republic at the University of Life Science. So welcome. And yeah, floor is yours. I think maybe you can put it in.
I want to hold it.
Yeah.
Yep. Cool. Hello, everyone. Thanks for resisting. And I'm switching a bit from indicators. I'm presenting to you RecRed. This is our effort to collect data on species red list assessments at national level. So you all know the IUCN red list. This is a massive effort to have all the species assessed at and the conservation status. They have more than almost 150,000 species. So it's the everyone that's using global level distribution ranges will have used this kind of data. But as you can see here for this example of the Shaguarundi, this neotropical cat, these data are expert drawn polygons, which are for the entire range of the species, and they have one conservation status, right? So we're, yeah, so however, despite this, this range and the least concern status, like we can see there, the species can be either extinct or really declining in specific regions of the world. And so this is why we are interested in this other sort of data. And luckily, many countries and regions around the world have produced their own threat assessments, and they all usually compile this as national red list. There's also subnational red lists. At our group, we are interested in extinctions at local scales because we study turnover. But we have partners in our group that are, for instance, interested in being able to predict the conservation status of species in places where there is no data. And I bet you can also think maybe on case studies that this kind of data would be useful. So this is not compiled. IUCN has this data on the entire range of the species, but each one of the countries that has their local red list it's not anywhere. It's not yet out there. So this is basically our goal with RecRed. Yeah, so to develop this comprehensive, we want it to be open access, geospatial also database that will store species threat status at regional, national, and subnational level. And we've done this in two steps, two phases. First, something that was already done, that we focused on the metadata, and now what we are now working on, which is extracting the data. So basically, the first phase, which is related to metadata, we basically search for everything every source of red list out there in the world. We compile it, we enrich it, and publish it as a data paper. That QR points to the preprint, but now I think last week the paper was finally out in Nature Conservation. So basically, this is around more than 2,000 sources around the world. Yeah. And from 172 countries. As you can see here, like some countries like this work, yeah, like the US seems to have very few, but actually it's because their red lists are red lists of fauna, and so they have everything under the same red list. And some countries like Germany, they have like an order of an insect red list, and that very like a state level. So there's a huge variety on this. And what else? Sorry, this gets a bit like, yeah. So there's a lot of taxonomic groups. We kind of wanted to see everything out there. Almost 500 taxonomic groups, and they're red lists that go back to like 1975, but mostly all are from 2000 onwards, which matches like IUCN efforts, right? So yeah, so in the second phase, which is what we are now, we kind of dedicated a lot of time in building the database structure. And then we also created an extraction protocol. The idea of this is to make it easy for anyone joining the team to be able to extract data from sources and to have it in our database. Then the part we are currently working on and has started like this month is extracting the data. And we have two ways of doing this. One is manually, and the other is automatically. About it in a bit. Finally, when we oh, nice. Epileptic. Yeah. And finally, when we have all this data together, we actually will put it in a PostgreSQL database, which is geospatial, and you will be able to click and see all of this data in a nice way, hopefully. So here's our structure. You can access here like the entire structure of the database. Each one of these represents a table in a database, and it has all sorts of terms there. So basically, we have a source that may have many red lists inside. Each red list has associated allocation, but actually allocation can have many red lists. Yes, and then the red list, sorry, I didn't say, allocation can be a country, can be a state, can be a county, can be a protected area, or a custom region, what we call, for instance, the Carpathians that are spanning several countries. So then this red list table has a taxon assessment table associated. So each red list has the list of species with their corresponding status. So the list of species is also, we have like a taxonomy associated to it, so that we can control for like which kind of backbone, taxonomic backbone we want it to be, because some of them are very old, they might be outdated. And then each red list also has like a criteria used to establish the IUCN status. In some cases, it's not IUCN. And so we have to map this to what we are taking as the standard, which is the last version of IUCN. So just to show you an example, it doesn't go, oops, yeah. We can see here a source. So this is a red list from amphibians and reptiles in Uruguay. And from this source, one of the red lists is for amphibians, the other is for reptiles, and that is from this year, and was the criteria used is the last version of IUCN. So that is associated to Uruguay as a polygon, right? There's many fields in each one of these tables. I'm very simplifying here the structure. So then within that red list, there's many species. For instance, this one is a tree frog, and it has a critically endangered status. So that is actually an outdated name. So if we check the taxonomy and, for instance, harmonize it towards the GBIF taxonomy, we will have, you know, Boana albopunctata, which is the most, yes, how do you say it? Well, you got what I mean. the newest. So whatever we want to be, this can be variable, actually, any taxonomy. And finally, in this case, we are using IUCN version 3.1, but some countries have their own. For instance, they will have G1 means least concern. So that's what this table does. It's just mapping one to the other. So, great. I haven't finished. Okay. Do you want to go back to my? Because yeah, good, forward. So one more, yeah, there. Manual extraction. So I told you about the extraction part. There's two ways of doing it. The manual one, we have an Excel template, which has all the definitions of all these terms. It took us some time. We have a great team working on this, so that everything is like defined and standardized. So we have several tables. A person that is extracting a source will grab the source, fill out these 4 tables here. Then they will have to, if it's not the conservation status, it's not IUCN, they will have to map this. Then we have like a stage of citations in several fields to relate to the source and get attribution, licensing, and all this. Then last steps would be to harmonize taxonomy, which I find out that Catalog of Life will be very helpful for us, and even integrating with GBIF hopefully may be possible. And for the automated extraction, I invite you to go to Adam's poster, because it's he will be able to explain about EcoParse, which in a nutshell is a tool to extract species names and conservation status from PDFs, or yeah, mostly PDFs, images, and things like this. This is kind of saving us a bit of time. Everything is corroborated manually anyway, but it helps us a lot. So yeah, drop by Adam's poster. Next. So why is RegReg cool? Because it's a geospatial relational database. That means you will be able to say how many critically endangered amphibians are in the world. You can also say how many, you know, and I don't know, non-endangered species are there in South America. So all the connections with this table you can think of will be able to answer with this database. Also, it's because it's spatial, it's nice and visually possible to click and see stuff. Well, it's highly flexible because we can take different backbones in the taxonomy, because locations are also polygons. We are using geoboundaries as a which I think GBIF is also kind of, or I got recommended by GBIF as a potential source for spatial polygons. And then also this mapping of different conservation status, which is possible. We are borrowing terms from Darwin Core, even Humboldt Core, and it's all the most standard it can be in this sense. It's actually also compatible with National Red List, which I haven't talked about. And this is a Red List project that is part of IUCN, but it's not working anymore. So they have digitized some data, but their efforts are a bit on hold. So they are part of our partners, and we are working with them, and we will be able to put the data that is already in National Red List, hopefully in our data, and so on. We will be able to integrate both projects, luckily. And finally, I think the thing about having an easy extraction workflow means that anyone that wants to contribute data will be able to do it. Yeah, and okay, this is our group. As you can see, this is our group in Prague, in Czech Republic. It's a huge group, but we also have partners, some of them in Germany, Australia, France, and here are the people from National Red List. Sophie Ledger has been extremely helpful, actually, in designing the database. yeah, so if you're interested in this, whether it is like you want to contribute with data, use the data, you're interested in the protocol, in the structure, just get in touch with us. We have a project on GitHub. We are putting everything there. The data will be there, the protocols. Now it's just the metadata are there. So with that, I'm done. I'm happy to take your questions. Hopefully.
Andrew, maybe you can repeat.
Oh yeah.
You can repeat.
Okay. Yes.
So thanks, that's super exciting. So I'm a red lister, so and now GBIF will get requests on how we integrate national conservation statuses with taxonomic names. So these are my first one. Is the intent one of the intentions of this project to then sort of publish this on GBIF? Okay. And then the second question was, having experience in red listing at national scales, I know that they're not necessarily comparable. And so coming from user perspective, how do you deal with these kind of differing methodologies?
Yeah, so first question about maybe potentially integrating this with GBIF. I think after Alice's talk, I was about fragmentation of data. We were thinking, yeah, this is something that could be actually in GBIF, as there is the invasive checklist per country. There's already this, I think it's called GRIS. And I think this could be a way. I'm not sure how, because we have statuses, right? And this is not a term for Darwin Core, but it can be a dynamic property maybe. We can talk about it. Yeah. So that's one thing. And the other thing was, so it's huge. Yeah, there's a huge variation in related to the different methodologies used to create this red list. Actually, some are, for instance, government documents. They're not proper red lists. Some are red books. So yeah, there is a huge variation. I guess we will have to deal with that when we see the data, how they look like. But our idea is that we will have a mapping table. So that's going to be part of the structure of the database, so that you will see that the verbatim status is one, but then it's translated to a standard according to what it's to IUCN. And this will be, in some cases, will have to be mapped by us because there's no reference. But in some cases, like the US and Germany, they provide already a reference to map this. They say, okay, this X3 is actually endangered. So if there is a reference, we will follow it. If there is no, we'll have to deal with it and come with a transparent way through a table to say this.
Thank you. So I think we have time for one more question. Yeah.
Mine is a small clarification. Are you picking only the species and the conservation status, or you are picking food assessments like we see in the IUCN portal?
This is good, thank you, because actually NRL is doing more detailed stuff like threats. What are the threats for the species? Not only like, oh, it's endangered, but also this and this and these are the threats. So I think this is something that could be added to our database pretty easy, but now we're not focusing on that. So we just have the species and the conservation status and under which criteria that was done. But yeah, there are, for instance, NRL has also like something related to if it's a marine terrestrial species. I don't know, some other little things there. Oh, we do consider actually the criteria. I forgot about that.
Yeah, because we're not extracting like sections describing the threat, but we want the criteria, the code that is like A2, C, D, 1, 2, 3, if you know, and that itself holds some information about what the threat is.
Thank
you. I'm sorry, we need to move to the last presentation, but I think you can approach the speaker after the session. Okay, so thank you. And we welcome our last speaker. He's Jerónimo Rodriguez. He's a Colombian researcher based on Washington, D.C., I believe, or I also see University of Minnesota in his affiliation. So Jerónimo will talk about characterization change, a time series approach to NCP mapping. And I think NCP is like natural capital, or you can tell us more. So welcome.
Thank you, Lina. So hi everyone. Yeah, as Lina told, I was based on WWF in the United States, but I just moved, and from now on I will be based on WWF Colombia, doing the same thing. So I'm back here, and my presentation is about characterizing change, a time series approach to nature contributions to people, and where is the here. So the goal for us is to create a global time series of key ecosystem services for all using monitoring and assessment processes. For example, IBBS global assessment, GBIF monitoring, and other global and approaches to this to following and to monitoring the change. But also, it is very important to see how this change are distributed across time and space, but also how we can combine this with the socioeconomic and demographic data in order to identify who is affected most by this change. And also develop a replicable pipeline to use for modeling efforts, for example, the Global Trade Assessment Platform and others that can contribute to integrate these contributions of nature to people into global, regional, and national accounting systems, because we are interested in integrating the provision, but also valuing it and who is affected by these changes. So what are our research questions is what have been the trends over the last 20 years globally, but also by different contexts, socioeconomic, regional, sub-regional, and who is most affected by these declines, what demographic contexts correlate with change, if there is some clear pattern that can be identified, and where are the gains and losses in this provision, spatially and socially distributed. So, drivers for change, so far we haven't not been able to identify a specific or causal relationship between the socioeconomic variables that we estimated and these changes, but what we can assess so far is the pattern. So how does this provision of services vary across landscapes? Is it possible to detect regime shifts and inflection points? Who are gains and losses? Where are they? And how are they spatially and socially distributed? And in this way, inform decision-making process. So for this, we used a set of globally modeled ecosystem service layers in the raster format, and also spatially explicit gridded representations of beneficiaries or different layers of demographic data. We extracted, this was originally based on the ESA land cover map at 300-meter resolution. What we did is to summarize this on a global 10-kilometer grid that makes 2.5 million cells, and for two different dates, originally 1992, 2020. In the next steps, we are going to fill the gaps between the two dates, initial and final date, and estimate the bitemporal change in this provision of services, and extract both the absolute and the relative change. For this, in order to extract hotspots of change, so the places where the change has been the most, in this case, we were interested in gains of either in increased damage, so increased risk or increased pollution, or or reduction in the service provision, for example, pollution absorption, or sediment retention loss. So the output of this is a graded cell, like the same graded cell input, but with values at each cell level and identifying the hotspots. And the idea was to extract, or the objective is to extract if there is a pattern or an association between the amount of change and who and where are they affected. So for this, we use data produced using the integrated evaluation of ecosystem services and trade-offs developed by the Natural Capital Project at Stanford. Extracted summary stats for each one of these grid cells, calculate the bitemporal change, and identify these hotspots. Finally, it was mapping and graphing and doing some statistical tests to see what kind of patterns can be identified here. Those were the input layers. So, and these layers are the ones that we have been able to model globally for the 2 dates. It's depending on the nature of the service. In some cases was the sum for the grid. In other cases was the mean, depending if it's an index or an absolute value. And these are the global layers of demographic data and biophysical or socioeconomic data that we try to relate between these two layers. So here are some water-based services for the initial date, for the second date, and as you see, there is a small change, but at the global level, it's not very easy to interpret, okay, what is happening. So what happened here? This was also, so we developed the, in terms of nature access, nitrogen export as a proxy for pollution, nitrogen retention, and also the provision of the service, which is the ratio. In the same case for sediment, universal soil loss equation, and to obtain also the value of the service and also pollination. So going forward, those are the water-based services for each date. Then also pollination and nature access, which of course depends heavily on land covers and uses and the distribution of farms and population. So people within an hour of a piece of nature. So how has it changed over time? Assuming population consistent, and also coastal protection, not mapped here because it's just a thin stripe on the coast. And those are our layers of beneficiaries. One date, the latest date available to see, okay, how this change is related with the distribution of the users, the beneficiaries, or the amount of economic activity happening there. So again, a threshold of change of 5% in some services is the loss of the service provision. In others, is the amount of the damage created, for example, by pollution. Looking for the top or bottom tails, depending on the service that we are assessing, and comparing the hotspot to the non-hotspot to see if there is some significant difference or not. So these are the top 5% for each one of the services we assess, the amount of relative change in each case for sediment retention ratio, pollination, and these already reveal some patterns of where, independently of the absolute term, where the most change has happened between the 2 dates. So also, we can aggregate these services because they have been brought to a common standard, to add them together and to see what locations has more change in most services. So this is very useful to prioritize, to compare, but also to prioritize policy interventions, and combined with different combinations. In this case is the damage or the loss of access and pollination, or in this case, the provision of the service like nitrogen retention ratio or sediment retention ratio or coastal risk reduction. And we already see like some places in the world where like this change is concentrated. And what we wanted to see if there is some kind of specific or identifiable difference between these two hotspots and the rest. Are they spread out, or how are they distributed over time? So we tried different visualizations and representations of this change, comparing the two, comparing like each service with the beneficiaries demographic data. This is for each one of the combinations. So one side is the non-hotspot and the other side is the hotspot. So how broad, how spread out are they, or how they compare with the non-hotspots. In some cases, the difference, like they seem to suggest like very normal distributions, but, and this is for in each case of the service, but because this was not telling us a lot, we drove a lot a series of Kolmogorov-Smirnov tests in which we compared the two samples to look after, try to identify consistent differences. And what it shows is, depending on the service, there is a very significant difference for some of them, like this lighter colors, which is, for example, nitrogen retention ratio and GDP, but in others, the difference is minimal. So we see here like the differences, the distribution. So lighter colors, broader bars mean that there is a larger difference, like there is a distributional difference between the population or each specific variable and the existing provision of the service. And finally, this represents the empirical cumulative function between the hotspots and the non-hotspots. In some cases, it's very clear that they take lower values, but in others, the difference, there is no difference. There is no significant difference. So we need to go one by one and understand why and in which cases this demographic socioeconomic data is related with the change or not for each one of these different combinations. And finally, what we see is that in some cases, there is a clear difference between hotspots and the backgrounds. And this distribution between difference between grid cells across the different data sets says the direction is consistent for many services. Some are tied for risk reductions tend to be in places where are more densely populations. In other is the contrary, but this allows to focus. Is expected to allow to focus and prioritize better the responses. And this is so far the current status of this research. Of course, this can be also reduced at a regional level because the grid already exists, and I'm happy to take any questions.
Yeah, so there is one question.
Gracias. Thank you, Jerónimo. Very nice work. I'm just wondering if we can just take this global map and kind of down scaling to a country and that's it? Or you have to process?
People online will not listen if they are not.
The final data set includes an individual country column identifier. So this synthesis can be done at any level from regional, sub-regional, continent. We have also columns representing OECD or non-OECD member income levels. So all these comparisons here are at the global level, but we can run them at any level. I forgot to add here the GitHub repository where the whole methodology has been developed. But yeah, this can be done at any level right now at 10 kilometers, but the methodology also allows to run the synthesis and the summaries at any spatial scale that the user wants. So if you have a 10-kilometer grid, that works at a 10-kilometer grid. If you want to do it by basin, by administrative unit, also it is possible because the workflow already has taken care of that.
So we don't have more questions in the room, also not in the chat. We continue with the discussion. We have 10 minutes for discussing But before that, I want to thank all the speakers for being in time and for the presentation. Yeah, thank you very much. So, like, first, we also want to hear from the audience here and online, and maybe to have a reflection of what you have seen and what tools could be used, and also what could be the role of infrastructures like GBIF, GeoBond, for supporting how we can translate all the tools and data that is becoming more and more available for the reporting. And I think this question is also, I think it's like very common that scientists speak to scientists, but we don't have that much communication. And I think that's also something that we need to improve and reflect on. So maybe if you have some ideas, if someone wants to start. Okay, we have one person.
Thank you. My name is Reem Al-Maalla, and I come from Bahrain, which is a country in the Arabian Gulf. And I'm both a scientist, but also work in the policy world. And I lead the GBF alignment with the National Biodiversity Strategy in Bahrain. And so one of the challenges that that we face, and one of the things that I've seen being here, at least there's 3 different talks that I resonate with, but also feel like we can add data towards, for example, the red list, the red rag talk, because we worked on a national red list and we updated it, but the data is just there with us. And we see this trend where certain species that are threatened locally, but on an international scale, they're completely fine. And we only have three pairs, like, you know, and then they're going to be extinct. And on another level is bringing all the data together. So in terms of having like an actual structure has been a challenge. So we tried to build something with the CHM when countries were asked to come up with their MBSAPs in 2015 under the CBD. And then we were trying to come up with a database at the time, but there was a big challenge in the finances to actually pay for the cloud to put the data. So obviously then we were offered the infrastructure through the CHM portal in Belgium. But then the CHM is now considered invalid. Well, not completely invalid, but like, you know, it's gotten to the point where now people are migrating to other portals, shall we say. And it is that race on how do you collate your national data, digitize it, given that it is a different sources, different languages, you know, trying to standardize it into international, so that international standards, so that it's comparable. But at the same time, try to convince both scientists and the governments to keep it open access. Because again, there's so many hoops to get through. So the question remains as to where does the priority lie? We've got 23 like international goals under the CBD, like the GBF at the moment. And on a national level, it is a challenge to address, shall I say, if we only take 50% of that. So how do we, like, it's a big question, obviously, there's no particular answer, but I do feel like being in spaces like this, it opens up certain, shall we say, pathways that allow us to identify where we can put our data. Because personally, I can already see areas that I could contribute data from both a scientific perspective, but also government.
Yeah, so we have another opinion. I think what you said is, yeah, quite remarkable. And I think also one of the issues is like how we can, like the infrastructures, how can support this transition, no? When some platforms are changing and how we can standardize the data. So.
Thank you very much. I'm Patricia Cole from Conavio in Mexico, and I apologize I couldn't be the whole session because I needed to be in another one. So, but what I've seen is that in many countries we need to use sometimes global data when you don't have enough data, because we always need to have a more find data, because 10 kilometers, for instance, like it is huge different ecosystems in countries with Colombia, like Mexico with the orography. So just in case we don't have data, I would use that kind of data because I see the pollinators, we don't even have the complete list of pollinators, neither the interactions to see this is the hotspot. So we really need to take that into account as well as for the restoration program. You know, we are building one system. I'm going to present that tomorrow, by the way. But they want us to report and to see how the goal is fulfilled. And restoration takes like 10 years, if not 50, in tropical forests. So we need to be aware that the timeline is not just, I'm doing this kind of restoration because it will take time. So just, I agree with what you said, and we need to be aware that the goals are quite important, but most important is the decisions the countries need to take instead of just trying to look well at the CBT.
Yeah, that's a very strong reflection, right? Because the purpose is not just the indicator, but the outcome that is really in place. Do we have more comments, opinions? Yeah.
Thank you. My name is Vincent. I worked at Biodiversity Observation Network in Quebec, in Canada. And we've been integrating many data sources for a few years, developing our own infrastructure and consuming both local and regional data sources that are from like endangered species lists or observations from local partners and global sources. And we develop indicators from that. So I really appreciate like the whole landscape of what we had in the session today, like from those data sets and to the indicators. One challenge that we found in using global data sets is that some data sets are really well maintained and infrastructure are well financed, like GBIF. But also a lot of those data sets are not that well financed and they tend to expire and not be up to date. And so we have the challenge we have to report to locally to the regional governments. But there's also this wish to report to globally to be able to compare our attainment of targets to the rest of the world. And so this tension between local and global data sets is very real. And I really appreciated the presentation to try and tackle these challenges between like regional data sets and global. But I think that there's no clear answer to that. The big one, the answer that we have implemented is that we have a team and financing that is big enough for us to work in integrating like local data sets and be able to compare that to global data sets. But I'm aware this is like a really exceptional situation. And so, when we compare like our indicators from local data sets and the same indicators from global data sets, we see the difference in values. And so this is an important challenge, and I really appreciated the experience of everybody today.
Yeah, thank you for the comments. Yeah. We have something online? Okay, I will just check what is online. So, okay. Oh, it's Temitope. I don't know if she's also the speaker. So I'm going to read what she typed is, I think we should always keep interoperability in mind, not only in terms of data standards, but also in the methodological approaches that enable platforms to connect and exchange information. When working with countries at the ground level, it's important to have mechanisms that allow data from different sources to be brought together, analyzed collectively, and structured in ways that make the results clear, comparable, and easier to interpret for decision making. And method to collect data on restoration site. Yeah. I think that's it. Yeah, thank you very much. Yeah. I think it's also a clear call how we can connect better with the local needs, because, and I think it's also connected to the previous comment, like we have these multiple scales, global scale, regional scale, national scale, and then the people on the ground are making the decisions, and let's say restoration efforts as an example, with data that is coming from these multiple sources. So there's a big challenge. We don't have much time, but I just want to know if there is someone that has a last comment. If not, we just can wrap up. I don't know, Andrew, if you want to add something? Yeah.
Yeah, I think I really like Patricia's comment around just we have to be pragmatic about this, basically. So often we go into these discussions, and one of the big things that comes up is, oh, yeah, we have these global data sets, we can't use them locally. When actually sometimes you can, sometimes you can't. But I think we have to be pragmatic in making sure that because ultimately it's about making the right decisions about biodiversity. I think one of the things that we are as infrastructures is to support some of these local needs as well. So we're talking about how using infrastructures to support the data sharing. And I think we do play a key role of that. And us being one of the nice things about GBIF is we are a government membership. So we really try to support our government members to be able to do some of that. But yeah, I think one of the things that actually really coming out of this, which I think is really positive, is that I think there's often a lot of negativity around sort of the availability of data for biodiversity reporting. And actually what I've come out from this session is actually there's quite a lot out there that exists already. And actually that has come out for me as being quite sort of positive part of this. But yes, I think being pragmatic about the data and building on the infrastructures that are there.
So yeah, thank you everyone. And yeah, I think we will be around. I think this is a very important topic. So you're welcome to approach us if you want to keep the discussion. And tomorrow we are again here, just remind the same room, Tolima, and the same time, 10:15, for the second part of this session. The last information I received, there are some lunch boxes waiting for you at the end of the corridor. So when you go out, take the right, and then you will find it. Thank you very much.